So I'll be talking about results from the COCO trial today. This was a trial to test the effectiveness of multimodal lifestyle interventions in Alzheimer's disease. And of those interventions, probably the most important are changes in diet and exercise. So this trial represents the blossoming, the flowering of a couple of strategic initiatives that ISB has had, one pretty much for the entire lifespan of ISB, to implement systems biology as an approach to translational clinical medicine to discover treatments for medical conditions and improve wellness through systems biology. And the other is to test to see whether or not we have an effective treatment or amelioration or even cure for Alzheimer's disease. So these two threads or these two strategic initiatives to some extent are very different kinds of knowledge. So in one case, I'm trying to come up with a new way of learning. So an approach to learn more about medicine. And in another case, I'm trying to apply that approach to learn something. And so those are two different threads, and I'll be sort of switching between sort of this philosophical epistemology, how do you know what you know, how to learn new things, and also the practical results of applying that. What is it that I've learned? And so those are the blossoms on this tree that are beginning to fruit for us with the results of the COCO trial. So switching back to my presentation now. So the first of these strategic initiatives is to think about biology and think about humans, think about medicine from a systems point of view. And typically when we think of systems and we implement them in terms of actual learning, we implement them in networks. But in this case, we're talking about multi-scale networks. So networks of networks, networks at the genetic and the molecular scale, all the way up to the social scale. Now, most 20th century medicine, when in the 20th century, when we're looking for drugs, even today, when the FDA and big pharma is looking for drugs, they're almost exclusively focused on a single plane of these networks, these molecular networks. Often, for example, looking, say, can we find an inhibitor of a specific enzyme? Can we find a particular antibiotic to treat a drug? Can we find a new cancer drug? And it's been tremendously successful, but it often ignores all of these other levels that we think may be important for understanding and finding good treatments for complex diseases like Alzheimer's disease. The COCO trial in particular is very much a multi-scale systems trial. Our intervention for COCO is a social intervention. We call up the individuals, we code with it with the trained professional coach, but basically it's a telephone call. We call them up using their social network, if you will, and say, hey, you should make some changes in your life. You should change your diet. You should exercise more. You should do cognitive training and a few other things. That intervention, we hope, cascades all the way down through all the networks of the individual, impacts molecules in their blood, which then hopefully, if we've told them to do the right things and they're doing them, improves their cognition by influencing their organs, their brain, and not only improves their cognition, but also their function. So it's very much a systems approach to thinking about medicine and a systems trial. So how do we actually implement that in terms of knowledge? This is a bit of a metaphor. One of the things we do is measure lots and lots of things in the same way that with a digital camera, you're measuring many millions or thousands of pixels, and together all those pixels create an image. So in this case, if you're using a really poor digital camera, maybe you're only measuring 20 things, which is exactly the way clinical trials were done in the 20th century, you might sort of be at the top end of this scale where you're seeing only 10 or 20 little dots, the pixels, and you're pretty much limited in the kinds of insights, the kinds of questions you can ask. You can say, is this particular dot getting more pink over time? Or how many pink dots are there? And that's actually what gets you FDA approval. They say, okay, yep, check, this drug gets you more pink dots, you're approved, you can use it to treat cancer or whatever. However, once you get enough digital pixels on your camera, instead of seeing individual dots, by the time you get near the bottom of this, you're no longer thinking about these squares as individual dots, but rather you're perceiving the whole flower. And instead of asking questions like, is this dot getting more pink? You're asking questions like, should I water this flower now? And the reason I make that point is the kind of question you're asking is not just a question of scale. There's an emergent property that changes, and it's subtle. It's not exactly clear when you start asking about watering the flower, but it's a very different type of knowledge and a very different type of question that we want to introduce to 21st century clinical trials in medicine so we can learn more from the dense data that we're getting. I'd like to point out one of the reasons we can do this is it's become a lot cheaper in the last 20 years to get the kind of data where I can basically measure all of the molecules in the blood. So I can do this now where I couldn't do it before. Nevertheless, this actually has been done before. What I'm talking about is the kind of knowledge that naturalists in the 19th century used to generate knowledge. So this is these are pictures of Alexander Humboldt and Charles Darwin, both of whom sort of explored the world and used all kinds of systems insights to generate some of the most important insights in biology. Unfortunately for them, at the same time, many of their contemporaries were snake oil salesmen. They were recommending people take things like mercury to treat eczema. And so we absolutely needed the rigid statistical framework of the 20th century to figure out what was snake oil and what wasn't. Unfortunately for these gentlemen, we more or less threw out their entire approach to knowledge, which now we're trying to sort of rediscover, reinvent. And in fact, in some sense, we're only recently enabled to do that for the molecular systems of the body, because unlike, we can't wander around the Amazon unless we have the tools and the ability to measure all the molecules in the blood. And so the world has changed and we're trying to take advantage of that. What's a little bit ironic, however, is because we've been doing things in one way for 100 years, this 20th century linear, rigid statistical approach, we're now in a community of scientists where there's a lot of pushback against systems knowledge and the kinds of approaches to epistemology and knowledge generation I'm presenting today. And strangely enough, and the irony is the most important medical fact of the 20th century was done with systems knowledge. And that fact is that smoking causes cancer. You'll find many people today that I'll present, at least scientists, people on the street are like, yeah, no, we get it. It makes complete sense what you're doing. But science said, well, you can't do it this way because this is the way we've always done it. And those same people will embrace this bit of knowledge that smoking causes cancer. What was it that allowed us in 1950, right in the middle of the 20th century, to make this claim? And it's more than a claim, it's considered knowledge. And it's really 12 pillars, and I've got them in sort of red boxes around here. In this particular representation, it's 11 pillars. Some people split them up. And most of the FDA trials for drugs rely on only 2 of these pillars, sometimes 3 or 4. And those are these pillars down here in the lower right of strength of association and significance. Some of you will have heard of a p-value. That's significance. A lot of biology in the FDA is driven by this concept of a p-value. In some cases, they also use dose response and time dependency. But what we really want to do is build knowledge out of not just those 4 pillars. We certainly want to use those 4 pillars, but all of these pillars. And notably, systems biology really leverages these 2 pillars over here on the right, biological plausibility and coherence. And what these are is biological plausibility is building on all the other research that everyone has done, the millions of papers that are in research archives, all of the accumulated knowledge, and the world is actually changing very fast. There's been a lot of knowledge in the last 10 years that we can build on and integrate our new findings with. And the second is coherence. And that's where this metaphor I showed you of the digital picture, where we're taking thousands of pixels and it's showing us a picture of a flower. So as we generate new data, we're not just considering each little pixel in isolation, but we're seeing a coherent picture, and we're using biological plausibility to build that. And so again, just to reiterate sort of what I mentioned at the beginning, there's 2 themes coming together in this talk. One is we're trying to build knowledge in a new way. And the other is we're trying to test to see whether we can find a cure for Alzheimer's disease. By cure, I mean, can we slow it down? Can we make it better? Can we reverse it? Can we delay it? And so together, those are all I would consider ameliorate Alzheimer's disease. And in the extreme, it would be a cure. I want to be careful of using the term cure, but if we can make it better, I'm happy. So by now, some of you are saying, that's really cool. You've just created a complicated new way to generate knowledge, but really, do we need it? After all, I am here, I'm the FDA cow using 20th century principles, and I'm picking all low-hanging fruit from the tree. And it's been incredibly successful. We've been on a great run. We found lots and lots of drugs over the last 100 years, and we can totally see continuing to do this for the next 100 years. There's still a ton of really cool drugs just hanging on the tree, no reason to use systems biology to find anything else. And that might be true. But of course, ISB's job, in part because we've got to be small and nimble and creative, is to challenge conventional wisdom. And so we have a hypothesis that maybe, just maybe, all these apples in the tree are not the same. Some of the apples at the higher levels that we're not even trying to pick right now might actually be sweeter than the low-hanging apples. And so what if the best cure for Alzheimer's disease happens to be an apple you can only find with new approaches for knowledge? And so that's one of the reasons why we're doing our trial in our way rather than use traditional approaches. And I can tell you right now, the big pharma is totally picking apples from the bottom of this tree. So we get to see our approach side by side with their approach as we move forward over the next few years. Why do I think we need to pick apples higher in the tree? Why do I think Alzheimer's disease is a complex disease? Perhaps, maybe there's not going to be a single silver drug bullet that's going to cure it. My hypothesis, my model, our model at ISB, all our team's model, is that Alzheimer's disease has multiple causes. So, for example, it can be caused in part by synaptic dysfunction. You've heard of protein aggregates like amyloid and tau. It can be caused by early life viral infections like herpes viruses. I'm almost willing to guarantee you that COVID will contribute to the development of Alzheimer's disease in the future. It can be caused by genetic factors in some people, and it can be caused by eating too many donuts or sugar or carbohydrates. And here's the thing, you can have two people with Alzheimer's disease. Each of them has multiple causes, and yet none of those causes are the same. So it's a very complex clinical setting. And therefore, we hypothesize it's going to take multiple treatments, and those treatments are going to have to be personalized. The same set of treatments is not going to be the best treatment for everybody. So you're going to have to figure out what's going on with each individual separately. And we think those treatments may include traditional drugs, exercise, diet, cognitive training, and probably some other treatments, maybe even some that I haven't even conceived of. Now, once a person has a set of causes and we give them a set of ameliorative interventions or treatments, I think, and this is my hypothesis, that at least some of the effect of those treatments are going to be mediated by molecules in their body, specifically in their blood, and that those molecules, and you may remember the systems medicine slide I showed you early, are ultimately going to impact the outcomes in those individuals. So in particular, whether or not their cognition is improved or at least does not decline as fast, and also other outcomes like can they function, like go shopping and those sorts of things. And so I'm going to look in the COCO study not only to test to see whether or not I can improve cognition and function with multimodal treatments, but I'm also going to try to figure out why. I'm going to try to figure out the mechanisms, and that ultimately I think is going to be a very powerful way to do science, because at the end of the day, I will not only test this simple hypothesis, well, relatively simple hypothesis, but I'm also going to learn something about Alzheimer's disease, which is going to lead me into the future and enable me to make whatever treatment I have now even better in the future. But I'm getting ahead of myself. Let me just mention for some of you who may be thinking already deeply about this, you'll notice that there's some pentagons here. I'm hypothesizing that some of the molecules in the blood, these triangles, are direct mediators. That, for example, there's something related to what you eat, which then it directly influences something in the blood, which then either directly or through a chain of events leads to better cognition. There may be some molecules which are also altered, but don't directly lead to the final outcome. And those I've drawn sort of as pentagons here. Some people would call them biomarkers. But they may be highly correlated with the actual mediators. And for purposes of this talk, I'm just going to lump them together and call them mediators too. And you guys can ask me more questions about that later if you want. So if I want to prove, actually, let me step back. So just to reiterate, for those of you who did not go to my talk a year ago, what we're doing is we're asking people to change their diet, to exercise more, and to do some brain cognitive training. This is the slide that sort of resonates with that intervention. And then I'm following everybody for 24 months who's in the trial, measuring this deep picture of essentially all of the molecules in their blood about every 4 to 6 months. And then from all of those measurements, I'm sort of building up this picture, this metaphor of this pixelated flower that gives me the image of what's happening in their systems over time. So that's sort of the trial design, if you will. It's a 2-arm randomized controlled trial with about 25 people in each arm. It took a lot of institutions and people to do this. So this is a graph of the institutions that were working on the trial, included ISB, the Schenkel Clinic, and the Hoag, which are down in Orange County, California, were our main recruitment sites. We've recently teamed with the Pacific Neuroscience Institute to help us with some of the analysis and extensions to the trial. We leveraged either by contracting with or collaborating with a number of companies that provided various services. And the entire project, or most of the project, was funded by Providence St. Joseph Health as part of a large-scale strategic initiative, ISB as a research partner of Providence. And so the good news, and this is of course why I'm giving the talk, we're done. We finished collecting data on the last individual late last year. We've been analyzing the data for several months now. We still have a lot more analysis to do, but I'm going to give you a sense of sort of our preliminary skim, some of our top level results. And just as a tangent, and I really, this is worth sort of pausing and for a moment and give ourselves sort of I don't know whether it's a sigh of relief or a thanks or whatever, but this trial made it through COVID. Many clinical trials had to shut down and never recovered from COVID. And there were a couple aspects of our trial in retrospect that allowed us to survive. And these, it's well worth noting these because it could impact the design of anyone designing a trial in the future should take note of these, because there could be other unexpected events. There are almost always are unexpected events with a trial that's long enough. Firstly, our intervention was telephonic intervention. So for this population during COVID, we could not have picked a better intervention to deal with the need to do physical and social distancing. And the second thing was our willingness, our embrace of our new approach to epistemology, which is very general and can handle data in all kinds of forms. And it has 11 pillars instead of 2 pillars to drive it. And the reason is because many sort of FDA trials are straitjacketed by their design, and they require very rigid data gathering, often at specific time points, often synchronized. So as soon as COVID came along and inhibited their ability to gather particular types of data for months at a time, many of those trials just would belly up. Although we would have loved to have some of the synchronous data that we were planning on getting during COVID, we were able to adapt more easily. So having a robust analysis and being ready to think about our data in multiple ways was one of the ways that allowed us to survive. By now you're thinking, you probably heard me disparage the 20th century enough that you're thinking there's no way Jared's going to talk about 20th century statistics, but here's the thing. It's not obvious, but I was actually born in the 20th century. I was trained as a 20th century statistician, so I'm going to show you one 20th century slide. And this actually makes sense because I'm trying to use 11 pillars, all the pillars of knowledge, and some of those pillars actually are classic linear statistics. So here is a linear analysis of the cognitive outcome for COCO. And what you're seeing here is all of the COCO cognitive measurements from all the individuals over 2 years of time. And the dotted line is the linearized aggregate trajectory of everyone who received coaching intervention, and the solid line is the controls. And what you see is a gradual decline in the coaching individuals, but an even more strong decline in the standard of care. And this is a significant difference, and it's also a fairly strong difference. So for example, for those of you paying attention to like the literature in the New York Times and the big pharma companies that are the cow picking, the apples from the bottom of the tree, the most recently picked apple is a drug called lecanemab. It's getting a lot of press. It's going to make somebody billions of dollars, or at least the company billions of dollars, the investors billions of dollars. And the big news for that drug is that it reduces cognitive decline by 27%. And just for what it's worth, and I'm not saying this is a fair comparison because of the nature of the linear analysis, but actually it's the nature of the business. The difference between the solid arm and the dotted arm here is a dotted line is about 66%. So our effect size is about twice as much as this new single mode drug that Biogen and SI are now promoting. And it suggests that since I'm not getting on the New York Times as often as they are, I need to invest in more marketing. Maybe I can borrow their marketing budget for a few days and rectify that. But anyways, we need to get our message out. So, but this is the 20th century result. And you're saying, wait, Jared, you're a systems biologist. what should you do next if you want to look at these from a systems biology data? So first, baby steps. One thing is to relax the assumption that things should be linear. I mean, biology is just not linear. There's lots of curves in biology. Things go exponential or they oscillate. And so maybe I should allow these trajectories, these cognitive trajectories, to sort of fit the data more naturally, not constrain them to be aligned. And if I do that, this is a spline fit to the same data. It's exactly the same data set that you looked at before. But by letting it become a little bit nonlinear, a couple things pop out. First, most of the difference, there's a big difference, between the trajectories early on during the first 6 months of the trial. That suggests to me that something substantial and important is happening early in the trial, and that maybe is one of the places I should focus a lot of attention on. And then after that initial, there's a sustained divergence which lasts 2 years. And that's also an important observation because a lot of drugs, like if you look at some of the early drugs for Alzheimer's, like donepezil, also known as Aricept, most of the effect of that drug fades after a year. So if I had seen a big effect but hadn't followed these individuals for 2 years, somebody might be able to say, well, yeah, but maybe it goes away after a while. It doesn't look like that's happening. So I'm going to focus next on some molecular analyses on the first 6 months of data. And I'll show you those in a second. And the other thing is this gives me an insight of how I should design my next trial. Like, I really wish I'd had a few more time points earlier because I didn't realize I was going to see a big effect around, but that's the nature of science, so I have to iterate. So it'll be important not to think of COCO as the final word, but to think of COCO as a bit of a learning experience. And the next time we do a trial, we're going to learn even more. Okay, so I may have convinced you, at least initially, that yes, the COCO intervention does delay or improve on average in aggregate across a bunch of people cognition. And I didn't show you my function slides, but we also see similar improvements in function. But now I promised you more than that. So I want to know not just the fact that there was an effect, but I want to know the mechanism. I want to know why, or at least tell me something more about Alzheimer's disease, right? So what I'm going to do is try to identify some of these molecular mediators, these triangles that mediate the causative chain of events between the treatment and the outcome. And although there's lots of ways I can do that, because after all, there's potentially these 11 different pillars I can look at. I'm just going to look at a couple of the pillars for now. I'm really going to focus on the biological plausibility and coherence pillars, some of the basic systems biology. And I'm going to do it in 2 steps. So if one of these mediate, if I'm trying to identify a molecule that I'm proposing to you as a mediator, it should have 2 properties. First, it should change in response to the treatment. Right. So if I give you the treatment, then the particular mediator should change in response to the treatment. And second, the cognitive outcome should change in lockstep with that mediator, or it should be correlated with that mediator. So both of those things should really be true for me to claim that I have a particular molecule which is a good candidate for mediating the effect of the treatment on the outcome. So I'm going to show you 2 slides. The first, I'm going to try to make the first point, and the second slide, I'll try to make the second point. So this slide has nothing to do with cognitive outcome. It's just looking at, well, it's looking at all 1,500 analytes that are looked at in the blood. So roughly speaking, there's everything in the blood. The colors you can kind of ignore, but just for what it's worth, the blue colors are the proteins, the green are the metabolites, and the red colors are clinical laboratories. Like it might be something, the clinical laboratory, example of a clinical laboratory would be a red blood cell count. And I've spread them out in the scatter plot. And the ones that change in response to the intervention are bigger. The ones that are most significant are bigger than the little ones. And so you see a couple things here. So first, there's actually quite a few analytes or molecules that change in response to the intervention. And the second is that they tend to cluster together, they're not randomly distributed. And so I find this really cool, right? But some of you are probably saying, okay, yes, this is not a surprise to us. If you change your diet and if you exercise a lot, of course, there's going to be a whole bunch of things in your blood that change. You are just stating the obvious by showing us this slide. And I'm actually perfectly happy to say, yeah, I'll grant you that point. This is an obvious slide. I've merely shown the obvious. It is a little bit gratifying because I think everyone who's not a scientist thinks this is obvious, but there's a whole bunch of scientists when we were pitching this project, we'd say, look, we're going to do this coaching intervention and it's going to cause the blood to change, it's going to make people better. And a lot of people said, nothing's going to happen. You can call people up on the phone and they're going to tell you their exercise is not going to do anything, right? And if they do anything, the people in your control arm are also going to do a bunch of things. You're not going to see any difference. So people do things when you coach them effectively. And there are big differences in their blood. Now, I haven't shown, this particular slide doesn't tell you whether any of these molecules influence cognition. It just tells you that if you do some major changes in your lifestyle, they will be reflected in the molecules in your blood in a big way. And then the fact that these are non-randomly distributed, it tells me that there might be some specific systems that are changing as opposed to just random molecules here and there. That again makes a ton of sense. If you're exercising, you can imagine various systems associated with muscles and energy use are going to change. And if you're reducing your carbohydrate intake, for example, there's going to be a number of metabolic changes as well. So it's not surprising that these are non-randomly distributed. Great. I promised you two lines of evidence. One, that I would show you some molecules that changed as a response to the intervention, and the other that there might be some of these same mediators which also are connected to cognition. Now, there's a number of ways I could kind of make that case. One is again, using networks. Systems biologists love network representations. And what I'll show you on the next slide is a correlation network. So this is all of these 1,500 analytes, or almost all of them. And then I draw a line between the ones that are highly correlated with each other. This is a proxy for them being part of the same system. So again, part of the same metabolic system, for example, that might happen when you reduce your carbohydrates. And I've colored in the next slide, I've colored in red all of the points that have two properties. One is they changed significantly in the previous slide, and the other is they're tightly correlated with a positive cognitive outcome. So the dots in red in the next slide are my main identified candidate mediators for conveying the effect of the COCO intervention on the outcome. And if by chance they cluster into these little subgraphs, that might tell me something more rather than this look at each individual pixel in my metaphor of a digital picture. But if they cluster, it might give me a sense of what systems are involved. So here's this next slide. It's not quite as gratifying as some of these toy pictures I've shown, but there is a sense that there's at least two clusters here. One is sort of a tight-knit cluster, which is a couple of immune molecules. And so that's a strong suggestion that there is an aspect of the immune system which is conveying some of the treatment effect of the interventions in the COCO trial. And then more sort of diffuse, but nevertheless clustered, are a set of molecules which could be conveying a number of different effects, but the message they seem to be conveying mostly is that caloric restriction or reducing your overall American eating too much habit is also conveying some of the effect. Neither of these are probably terribly surprising, but it's nevertheless kind of cool to see those effects. So in summary, what have I done? I've trailblazed, or we, it's a big team. We've trailblazed the future of clinical trials. This is, as far as I know, and I haven't carefully researched this, it's possible somebody has scooped me somewhere. I believe this is the first time anyone has ever done dense, dynamic, longitudinal data in a prospective randomized clinical trial. And I think it's been tremendously successful, even though we were gut punched by COVID. We lost, we have the total number of people on trials diminished quite a bit, but nevertheless, I think we've done a proof of principle trial. And I think we've also learned how to ameliorate Alzheimer's disease. So this is pretty cool, but you might be thinking, well, again, everything you're saying is obvious, right? Didn't we already know? You seem to be just told us that eating better and exercising are good for our health. And we're pretty sure we've heard that message before. We didn't need some big pharma wizard to go looking for some new drug. We knew this all the time. And to some extent, you're absolutely right, in the sense that I'm just telling you that diet and exercise are good for you. We've already known that. 10 years ago, there was a trial called the Finger trial out of Finland, where they did almost essentially what I just did, a randomized prospective clinical trial. They didn't get dense data. They were just testing to see whether multimodal interventions worked. And they found more or less the same effect I did. Just in the last month. I mean, you guys have seen I'm sure I don't need to point all these things out to you, because you've seen all kinds of studies published that diet and exercise are good for you. But the combination in 1/3 of a million people living in Great Britain shows that these people are living longer, at least in this cohort study, than other people. It suggests the combination is important. And that's one of the other criticisms we get for COCO is, why don't we test everything separately? Because sometimes combinations matter. Now, admittedly, this is about longer life, not dementia, but I think they're related. And then just in the last week or so, this was also kind of gratifying. A group did a purely theoretical analysis based on many of the graphs that I draw on as well. And they were trying to predict what intervention would be most effective for treating Alzheimer's. And out of all of the drug and lifestyle interventions they looked at, exercise was the single biggest factor. And so again, everything fits together. And this comes back a little bit to these 11 pillars of knowledge. I'm not just relying on my own data or my own analysis, but I'm integrating that to make with other people's research to get a context and a biological plausibility to bring home sort of this final message. And maybe I can counteract some of the messaging from the drug companies that, hey, also, we should be doing things other than drugs. In addition to drugs, these drugs are quite useful and maybe part of this multimodal intervention. So what are the next steps? We can use the insights we've gotten from this trial, not just to figure out how to make better trials in the future, but we can also tweak, refine, personalize the intervention based on some of the things that we've observed. We can tease out some of the aspects of the intervention that are most effective, and we can kind of double down on those, for example, in our next trials. More analysis is totally needed. There's so much data that came out of COCO. There's going to be so many cool things we can do. And, let's do another trial. And so we've actually already started. We'd love to do 10 more trials. If the NIH and other people want to fund us to do more trials, we will, but we've already started one. And this is the prevention trial. And it takes advantage of some of the insights we've learned from COCO, but not all of them, because we started it before we finished COCO. And in some sense, it's not quite as simple as this, but we're kind of doubling down. If some exercise is good in COCO, maybe a lot of exercise is even better. And so we're doing a lot of in-person interventions rather than just coaching interventions. And this is happening with our colleagues in collaboration with our colleagues at Pacific Neurosciences Institute down in Santa Monica, California. And we're wrapping it up, but it will probably be 2024 before I can give a similar talk on prevention as I've done with COCO today. Lots of people are involved in all of this research. I'll call out Raj Shankle, who's our clinical PI at the Shankle Clinic, who's done responsible for a lot of our point of care and running the trial down in California. Got pictures of some of our colleagues, both in COCO and prevention. We were recently down at the Alzheimer's conference in San Diego. And the most important people that never get thanked enough are the participants in clinical trials. These are altruists who are giving their time, much of their blood, for the benefit of future generations. And so I'm very grateful for all of the participants in the COCO trial. That ends my presentation, and I think we get to go to question and answer now.
Yes, thank you, Jared. You made an amazing case for systems biology, and I'm going to ask some low-hanging fruit questions first. You talked about diet and exercise. I think eating a good diet means different things to different people. Did you find any best practices for nutrition and exercise that had led to more robust results?
I'm not quite willing to answer that question, not in any situation where I could get quoted on it. The original design of COCO was to have 200 people participate, and we ultimately ended up with less than 50 who had full data sets. And there were a number of reasons for that. And so if we'd had a few more people, it would have allowed me to delve a little more deeply into what you might call sort of some of these stratification analysis. I still believe it's remarkable with so few people. So a lot of people think of a trial in terms of N, the number of people in a trial, and they think, oh, the smaller number of people, you can't get a lot of data out of it. N should be the number of data points in your trial. So with a dense amount of data, we can get a tremendous amount of data from this trial. And we're still working on it. We think that by tracing the paths, the causal paths or the mechanistic paths of particular analytes, particularly in the diet, we're one of the most effective places we can do this is with diet, because we know exactly how many of these metabolites are metabolized in the gut, the microbiome, and then are working in systems in the blood. And so we can trace the effects of particular dietary interventions. And we think we can get some more insight into answering that question. I can tell you though, just off the cuff, and this is, and there's an infinite number of diets. And so I think there's a lot more research to be done here. And it's exciting because I think this is more trials designed the way we've just done are the way to answer those questions. But off the top, just sort of the top level analysis, and this is confirmed by so many other research studies, at least for Americans, we eat too much. Cut down your calories and mostly your carbs, but also I think we're eating too much red meat as well. And I'm not at particular risk for Alzheimer's disease. Just being part of this analysis, I'm able to pick up that candy now and say, you know what, I'm going to put it back in the bowl. And I think we'll have a lot more nuanced suggestions for diet, both from our research and many other people's research. But I can't quite tell you that it's broccoli or spinach yet. Yeah.
Okay.
Vegetables.
Okay. Have you submitted for publication? I believe there's a preprint. I think we'll be able to post a link to the preprint for a publication on your preliminary findings. So you can confirm that. And then also, how are your findings so far being accepted by your scientific peers?
So we've just put our findings out as in a preprint, which means the entire world can see it. It's like publishing, but it's not peer reviewed yet. And I'll be presenting these results in a talk at the clinical trial. It's a big conference in December for Alzheimer's disease. I'm the last talk at the conference. The first talk, which is given full keynote speech, is like an hour long, is the talk from Biogen and SI on their lecanemab drug. And that's going to get all the press in the New York Times. And then by the time everyone filters out to catch their flight, I'll give my talk and to the 3 people left in the room. And who knows, maybe we'll get a little bit more traction there. I'm expecting some feedback as a result of that meeting. There'll be more than 3 people in the room. But, I've been there though at other conferences. And then we'll submit it to, we'll have the work peer reviewed. And there'll probably be a number of publications that come out of this. One of our problems is there's just too much to say. And most, you're only allowed to submit sort of bite-sized stories to research journals. And in fact, the link that you will send out in your email later will have 2 links. One is the short version, if you want to read a 3,000-word summary of what I just said, and the other is the 14,000-word details. So you could pick your poison from those links.
Okay, thank you. We're getting some questions from our audience. Someone asks, what if a pandemic happens again during the in-person trial? What will you do?
Well, to be honest with you, the pandemic, we were actually recruiting for prevention prior to the pandemic. So prevention got slowed down a lot. We're kind of learning on our feet a little bit. One thing that not only we, but almost all neurology trials, neurocognition trials, dementia trials are doing is increasingly relying on remote assessments. So prior to the pandemic, almost everybody's saying, oh, you need to assess people in person because people can cheat or whatever. I don't know, they're like high school students, or whatever. They've written the answers on their wrists. So there's new ways. People are really pushing hard to do things remotely. But yeah, our trial was slowed down, and we designed it before COVID, and we probably have designed it a little bit differently. We didn't shut it down. We managed to avoid shutting it down. But yeah, that was a problem.
What if somebody wants to participate in that trial? Is there a way they can find out more information?
Yeah, we can give you a link, I think, in the follow-up email, or maybe in chat. I can paste it if I get a chance. However, because it's got a strong in-person requirement, you more or less need to be within commute distance of Santa Monica, California. That's Los Angeles or Hollywood. I think it would be remarkably difficult to do that, for example, flying or whatever. But yeah, if you happen to be close enough to the Pacific Brain Health Center, also known as Pacific Neurosciences Institute, and if it's targeted towards people who are in their early stages of Alzheimer's disease. And I didn't spend much time because we had very small amount of time today. I never really defined Alzheimer's disease for you. Alzheimer's disease is a spectrum. When it gets really severe, it's called Alzheimer's dementia. Dementia implies that you're impaired in your activities of daily living and have severe cognition. But early on in Alzheimer's disease, there's more subtle signs. And so we're targeting people who are sort of in the mid-range. They don't have Alzheimer's dementia yet. They have sort of this mild cognitive impairment or even slightly earlier than that stage in the spectrum of Alzheimer's disease. So if you happen to be right in the sweet spot for enrollment, it'd be great. We'd love to have you.
Okay, great. Good to know. Someone else is asking, what role might alcohol have on Alzheimer's and dementia? Did you look at that in the COCO trial?
Nope.
Okay.
I don't know whether the person is proposing it as a treatment or a cause. Yeah, there's only so much we can look at, but I think that there may be ways to study that using systems biology in future trials.
Well, did participants have to track what they were eating or drinking? I wonder if that's something you might be able to look at that might be in the data that you just haven't looked at yet. So
the dirty little secret of diet diaries is there's almost no correlation between them and what people actually eat. At least this is what other people in trial studies have told me. Nevertheless, for prevention, we are tracking diet fairly carefully with these diaries. For COCO, we did not. Here's the thing. I think particularly because these diaries are not very effective. Now, maybe you could imagine maybe for these people that are taking pictures of everything they eat, there might be a way in the future to do it a little bit better. But by measuring everything in the blood, I have a really, really good insight into what you're eating, and you can't cheat. I know how much coffee you've been drinking. Now, there's limits to what I know because I'm not measuring your blood often enough to capture certain things that go in and out of your blood quickly. And that bothers me. I wish I could do a better job of that. And there might be some tricks. But I think that the way we've designed our trial, we're going to get better, more believable results by looking at what's actually in the blood, these xenobiotics, these foreign molecules that come from your diet that are in your blood, than by asking people what they've eaten.
Okay, thank you. Did you see any sex-specific effects in your study?
Yeah, we did. On that, I don't know what, again, I mentioned it a couple times during my talk. I was like, well, I see the results. It shouldn't be surprising at all. And yet it's kind of cool, or it surprised me. This is one of those, right? It should not be surprising. Like you ask anyone on the street, if you ask, on average, in aggregate, you average the results of a whole bunch of women, a whole bunch of men who've asked to go on a diet and exercise. Are you going to see differences in their blood? And most people can say, duh, of course. Because there's a lot of differences between men and women. Even simple things like their average size is different. But there's a lot of differences in the way their hormones respond to exercise. And the hormones then control lots of other molecules in the blood. And then the other thing is, and this is it remains to be proven, but it's probably true, we're using a coached intervention. If you tell a whole bunch of women, on average, doesn't necessarily apply to any particular individual, to do something like, you should exercise, you should diet more, and you tell a whole bunch of men, there's a decent chance that women are going to actually do more than the men. And that's what we see. So we see a little bit better effect in women for whatever reason, and we see whole bunches, the molecules that we see change are very different in men and women, and enough so that that's probably a whole line of research and publication we need to write up. That we just, after 14,000 words in our first paper, we're like, wait a second, we're not going to put this as a footnote in our first paper. This is a headline of itself, in and of itself.
Okay, thank you. Someone else asks, earlier in your presentation, you said you expect to see COVID as a driver of Alzheimer's, or at least a strong contributor. Can you explain your reasoning behind that?
So this is softer. I'm not in a position to prove, right? You've now entered the realm of we're just chatting, in a bar kind of argument. But there's a whole lot of diseases that have sort of been ignored until relatively recently, ranging from chronic fatigue syndrome to the various things that have been attributed to Lyme disease, to long COVID even, that And then there's also a number of other things like post-cancer treatment syndromes that share sort of cognitive dysfunction, even dementia, cognitive decline in common. So there's the things impacting the brain. Many of those can be attributable, at least speculatively, to earlier infections earlier in life with various diseases, various infectious agents, many of them viruses. Some of our colleagues here at ISB have been instrumental in showing that herpes virus can influence Alzheimer's disease. This is was one of these causative agents of Alzheimer's disease. So in other words, if you get infected with herpes early in life, that can increase your chances of getting Alzheimer's later in life. And just the mere fact that we're seeing COVID infections drive these long COVID syndromes with people with cognitive influence. And I think people have actually already, because people started to be able to look at cohorts that are 2 or 3 years old for COVID, are seeing an impact of previous infection with COVID on dementia, not necessarily Alzheimer's in particular. So it wouldn't surprise me if everything, I think everything is set up that COVID is likely to be a driver of certainly long-term cognitive dysfunction, if not Alzheimer's disease itself.
Okay, thank you. Do you still see a role for 20th century trials? You kind of debunked the low-hanging fruit apple picking. Are we still going to hunt for apples?
Yeah, the big pharma can pick all the low-hanging fruit, and we'll go after the high fruit, and it'll be synergistic, right? And in fact, I've been arguing for multimodal interventions, and I talked a lot about diet and exercise because I think those are the ones that are most impactful. But I don't actually see a particular reason. Let's say this drug, this lecanemab thing, does decrease it by 27%. It might very well be additive or synergistic with our effect, right? Can you imagine getting all the benefits of both? So yeah, absolutely. There's still a role. Although one thing I will say, and this bothers me to no end, the drug companies are solely focused on their bottom line and solely focused on the present. So they spend billions of dollars. So orders of magnitude more money than I had for this trial on their trials that they need to get FDA approval. And they don't generate dense dynamic data. It would only cost them a few extra $1 million to generate these data. And admittedly, their data wouldn't necessarily change whether or not the FDA approved their drug or not. But these people who are volunteering in their trials, their altruism deserves getting the maximum amount of data out of these trials. And if only the drug companies would also learn about Alzheimer's disease while they're testing to see whether their single one-off drug worked, tremendously missed opportunity, and that bothers me.
Well, I don't know if we have any pharma reps on this research roundtable, but we'll certainly forward some.
You don't piss them off.
Okay, I have another question for you. If you're testing 3 interventions, diet and exercise, and some cognitive, I guess, gaming that you're doing, some brain training. Are you testing the effect of them separately or the 3 intertwined, or can you tell which has produced a stronger effect, or is it really the combination?
So ultimately, we think we can use our systems data to tweak out the individual effects. And I sort of alluded to that earlier when I was talking about tweaking out the effects of particular elements of the diet. We're going to need bigger trials and more trials to fully get at all of these things. And there's there was a guy, a scientist, that just absolutely hated our approach to science and wrote this scathing attack on us in an editorial in the leading Alzheimer's disease journal. And we'll send that to you in a link. And then I'll also send you my response. But one of his claims, he says, it's just completely stupid to test more than one thing at a time. You need to test everything individually, one at a time, prove each of them work, and then you can start looking at combinations. There's a number of problems with that. So first of all, I have no problem with testing things one at a time. That's the low-hanging fruit approach. But if you do that, first of all, there's an infinite number of diets. So you're going to be at the end of the universe before you finish testing every single thing. And then you can start combining them. So we can't wait that long. We need answers earlier. So we have to figure out how to test things together. And then the other thing is, and I think I just showed you in a slide, I won't bring it up because I'm juggling, but we'll share this recording later. You can go back and look. That British study where they looked at 1/3 people. To extend your life, you needed to do both improve your diet and your exercise. You couldn't do one or the other to get that result. So yeah, I think synergy is important. I think systems biology ultimately will be able to deconvolute, maybe not all it's not like we're going to find the answers to every single thing, but at least some of the answers we'll be able to figure out. Yeah, it sure looks like this system. And I think I showed you in one of my networks, the system that was related to decreased caloric consumption, right? That's most likely a dietary effect. I don't think that's a brain training effect. I'm having a really hard time, for what it's worth, but we're working on it, teasing out the effects of brain training, because there's you could imagine that of all the things we're doing, that may not have molecular traces in the blood. And so that's going to be, with my particular trial design, at least as of now, we may have to future trials where we look at brain imaging and other things that may change this. I'm going to have a hard time teasing out that effect.
Okay. Well, we're running out of time, and I'm going to ask you a fairly obvious, easy question, and you're going to give me a fairly obvious, easy answer. And that is, if people don't know whether or not they're at risk for Alzheimer's right now in the present, should they still change anything about their lives now? I mean, is this information useful just for people with dementia, Alzheimer's, or at risk, or are your findings useful for everyone?
Well, the problem with big pharma drugs is they usually come with side effects. So these antibody drugs, like this lecanemab thing that I'm talking about, they have this side effect called brain bleeds. It's like, yeah, and making people really, you have to be really willing to risk your life, right, to take these drugs. But in many cases, it's worth it. So the problem with many pharmacological drugs, they have side effects. What are the side effects of making your diet and better and exercising more? Your side effects is better health. So not only, I mean, I'm just focusing on the cognitive outcomes and preventing dementia, but they also improve your cardiovascular health, they prevent heart attacks, they make you live longer. And so even if I'm wrong, even if you don't buy the fact that if you do what I say, you're protecting your brain from dementia, you're helping yourself in so many other ways. So most of us have a decent risk of dementia. There's double-digit chance that any of you will get dementia sometime in your lifetime, unless you're convinced you're going to die tomorrow and you don't have dementia already. There's a few edge cases there, but your chance, even if you quote unquote don't have risk for dementia, you do. And so I would totally go about, at the very least, cut sugar out of your diet. If you're drinking sugared beverages, sodas or whatever, stop. That's really easy to do. And then you can work on figuring out how to get rid of more sugar and more processed foods. And you can think about it. Some of you may not be exercising as much as you probably should. Walk around the block a few times. Just use this talk as yet another reason to live a healthier life.